Hello and welcome. You are listening to a podcast by the Milner Centre for Evolution at the University of Bath. I'm Professor Turi King, your host, and today I'm talking to Volkan Cevic, whose research focuses on understanding interactions between plants and their pathogens, as well as understanding how these pathogens evolve. So Volkan, how did you become interested in how plants and their pathogens interact? Was there a defining moment for you?
Yes, there was. I was actually trained as a plant geneticist. My study was on apples. And I was characterizing a specific phenotype in apples involving aphid resistance. And there are only certain apple varieties that can actually confer resistance to this specific aphid. And I was handling those individual aphids when I was inoculating apple leaves. But at the end, if the apple varieties are susceptible, they develop this beautiful red coloring on leaves, and the leaves basically curl. In a way, the aphid manipulates the apple leaves to create its own house, like a shelter, because the leaf curls and aphids actually go in there and they basically, live and reproduce. And I was really curious, how on earth, aphids actually sort of impose such, kind of changes in a plant species. So everything started that way. But then a few years later, with all this progress in plant field, then we learned what's actually happening. Similar kind of things are happening during plant pathogen interactions, plant insect interactions, plant nematode interactions. It's amazing. So there are common mechanisms involved in this kind of, obviously the components are different, but common mechanisms are involved in these processes in defining those interactions.
So Volkan, what kind of immune systems do plants have?
Obviously, plants do not have like mobile cells like we do, like mammals, and they don't have adaptive immune system, but they have sort of what we refer to as innate immune system. Of course, they have like physical system, those trees have barks to protect themselves, or some plants actually have thorns. But in terms of innate immune system, they have like two layers of immunity against invading pathogens or insects or other disease-causing organisms, I will say. So the first layer is very much effective against multiple pathogens. So what they do is they recognize what we call these highly conserved molecules. For example, what we know is in bacteria, they need to swim. So how they swim? They use flagella. So basically, plants evolved receptor proteins recognizing a bit of that specific part of that flagellum protein. That immune system is actually quite effective against multiple pathogens, because don't forget, there are multiple pathogens infecting multiple plant species at the same time, but only certain pathogens are successfully infecting only certain specific plant host species. So there is this beauty of this coevolution involved in this kind of process. But then, obviously, some pathogens evolved to overcome that immune system. So what they do is there are certain proteins in the pathogens or insects or nematodes. They basically secrete those proteins. And somehow some of those proteins find way into the plant cell, inside the plant cell. But then, of course, if the pathogen is successful in suppressing that initial immune response, then some plant species actually evolved another layer of immune receptors or immune system. We call it the second layer of immune system. So this involves so-called resistant proteins, but these are mainly inside the plant cell. So it's like two, layer of castle. So, if the first wall is demolished, then you have this stronger one. So you just want to protect yourself against all these invasions. So those proteins then, what they do is they recognize those effector proteins, which are actually translocated into the host plant cell. If there is no resistant protein within that particular plant species within the cell, then of course those effector proteins, they manipulate all sorts of different kind of processes. And they basically convert the plant cell into a food factory for themselves, so they can feed on them. Obviously, it's all about specificity. Yeah. It's all about coevolution, because, plants evolve resistant proteins against those specific pathogens. So those resistant proteins then recognize those effector proteins inside the host plant cell, and they then activate the second layer of plant immune system, which is unfortunately very strong, and that particular cell ends up dying. Don't forget, like plants do not have mobile cells or mobile elements in that respect.
Yeah.
Almost like every single cell is able to resist itself against invading pathogens. So what they do is all those cells infected by the plant pathogen, they sort of undergo cell death, and they actually enable to stop the pathogen. And that's a very strong response as opposed to the first layer. Because in the first layer of defense or resistance, plant cells are alive, but they produce compounds that actually enable them to resist against pathogens. This interaction is very specific. For example, we know that we have certain pathogens that can actually infect our roses, but they're not going to go and infect cabbages and like Brussels sprouts and things like that. There is a specificity. It's all as a result of coevolution. Then what pathogens do? They evolve as well to avoid that recognition. So they actually mutate their effector protein encoding genes, and then that particular recognition disappears. And then what plants do? They evolve a new resistant protein. So this goes on, basically. I'm really trying to understand that kind of mechanisms. What's actually happening? What's the diversity in the field? And can we have an ultimate plant immune system that can actually be very effective against multiple pathogens, ensuring for security in future?
And that's basically what you're trying to understand, but you're trying to understand it in particular species. You're looking at a particular pathogen, aren't you?
So my work at the moment focuses on what we call is white rust pathogen. So white rust pathogen infects what we call is brassica species. Again, brassicas are, everyday vegetables like, cabbages, cauliflower, Brussels sprout, but also important oil crops like oilseed rape and oilseed mustard. And especially oilseed mustard is one of the major vegetable oil crop in India. Basically, I've been trying to understand the infection strategies by this particular pathogen. And one thing I should mention here as well, white rust pathogen, it was actually considered to be a fungal pathogen, but it's not. It's actually another group of pathogen we call oomycetes. And it is actually related to potato late blight pathogen. That's the pathogen which caused Irish famine.
I mean, that goes into it quite nicely. What does white rust do to the plant?
Yeah.
So if it's very similar to this one that caused the potato famine, I'm guessing what it does is it really damages the plant.
Yeah. It doesn't cause such a damage like the potato blight. One of the things that we discovered throughout my studies as well as my colleagues' work is an amazing immunosuppressant. So basically, once it infects the host plant, it suppresses its immunity really effectively. Certain other pathogens come along, which will normally never infect that plant, starts infecting the plant.
So it's like it opens the door for other pathogens to get in.
Exactly. But of course, any infection will lead to reduction in photosynthesis. We know in plants, that's how we produce sugars and, how everything actually, is dependent on. That's one of the most important aspects of being a plant is the photosynthesis. It will basically massively impact the yield. But sometimes white rust can be systemic. What I mean by systemic is it infects, let's say, through the leaf, and it goes through all the, vascular system, and then it actually colonizes the entire plant. But then what it does is, after it completes its life cycle, it produces millions of spores. And those spores will be either through water splashes or wind. They will be traveling quite a long distance, and they can actually infect other plants within the field or nearby field or, further away.
So tell me, how does the white rust work? Because that's what you're interested in looking at, isn't it? You're trying to understand what's the interaction between this pathogen and this plant, basically how the pathogen is getting in, I'm guessing. Yeah. So tell me about that.
So white rust, they produce the structures called zoospores. A bit like bacteria, it has got flagellae as well, so it can swim. It requires water, basically, to infect. So these are sort of like water-dwelling organisms. They were actually considered to be fungus in the past because of how they look, but they're not actually. They're totally different. And they actually form these zoospores, and they swim, and this particular pathogen I work on enters through stomata. And then those zoospores, like seeds, they start germinating. In this case, obviously, zoospore will form what we call this hyphae. These are the extending structures of what we call these filamentous pathogens. So that enables to squeeze in those plant cells inside the plant leaf and colonize the leaf. But what is exciting is they form these structures, what we call this haustoria. So these are attached to the pathogen, hyphal body, and they basically engulf into the plant cell. So they push the plant cell, engulf into the plant cell, and they basically suck all the nutrients from the plant cell. Yeah. But of course, before doing so, as I said, they secrete and translocate those proteins. They go into the plant cell, they manipulate the whole plant cell, and they become like feeding factories for this particular pathogen. So, of course, one of the major aspects of plant cell is those cells are photosynthetic cells. They're active in terms of photosynthesis. They produce a lot of sugars. So what you want to do as a pathogen, maybe you just get all this sugar. You don't want to share too much with the plant itself. But, of course, this particular pathogen is quite exciting because at the same time, it can actually sort of systemically immunosuppress. So what it does is, it infects a part of the leaf, let's say one part, but the entire leaf becomes immunosuppressed. So even the non-infected part can now be infected by other pathogens as well. Just to make sure that plant has no resistance at all, basically.
So it's really taking over the plant.
Yeah.
Not even just where it's invading, but around it as well. So this is all about food security. There is a direct application for your research because...
From evolution all the way to food security.
Yeah.
Absolutely.
Yeah.
Absolutely.
Because eventually what you want to do is presumably be doing that for crops here. But as you say, you're already helping people in India. And so are the main crops that you're working on brassicas, or are you starting to move into any other ones?
I also started working on plant viruses. I have collaborators in Turkey and at Akdeniz University in Antalya. And that's one of the major areas where they grow glasshouse tomatoes and peppers and, vegetables. So during our conversations, they told me almost all the tomato varieties were infected by two diverse plant viruses. But what is fascinating is one of them, its genome encodes 4 proteins maximum and is threatening the world tomato production right at the moment. And it's amazing. How do you achieve that? How do you do this?
Okay, so let's back up a little bit. Viruses, what they do is they get into a cell, they take over the cell's machinery just to copy themselves and move on.
And then move from one cell to another, and then they actually reach the vascular system, and then they just move within the entire plant.
Okay, so what are you doing to preserve the world's tomatoes?
There are certain chemicals you can use, to kill viruses, but in an agricultural context, you are very much dependent on plant genetics, host plant resistance. So basically what you need to do is you develop new resistant varieties. That's pretty much, to my understanding, only way that we can actually deal with them.
Exactly. And the thing is we have an adaptive immune system. So we can give vaccines. Our body goes, that's a virus. I will make something against that virus and I will get rid of it. But you can't do that with plants.
No, absolutely not.
So you are basically stuck, as in you need to have varieties that are automatically immune to some extent, or where you do genetic modification, presumably.
Absolutely.
Where you give them that immunity.
Yes, exactly. But the problem with viruses is you develop a new variety, you just spend years to develop that variety, and then you put them in the field, and within a year, it's no longer resistant to virus because virus evolves so quickly. They've got this variance. I'm actually doing a different approach. In my lab, what we try to do is, as I mentioned, for example, in this particular virus, it's got 4 proteins maximum, but we actually focusing on 2 of those proteins and what they do in the plant and what sort of plant proteins they actually target, what sort of pathways they actually manipulate to make plant become a virus replication factory, basically.
So is that a way of attacking it? So stopping the virus from even getting into any of the cells? Or is there no way you can do that? You just basically have to give the plants some sort of immune system once they get in?
Virus will find a way because damages and things like that, there will be a way for virus to somehow enter the plant cell. Just one single plant cell is enough as long as it can enter into one of them. Yeah. But then what we can do is we can find some new mechanisms to stop virus moving from one cell to another.
Okay.
Or we can actually stop virus to go into the vascular system and to move within the plant. So that's what we're trying to understand now. So what we're doing is we're actually finding which plant proteins mediating that. It's like an Achilles heel, basically. There are certain host proteins. What makes us susceptible, for example? What makes humans or animals or plants susceptible to other pathogens? Because don't forget, those enemies, pathogens or viruses, they take advantage of your own protein or your component within your cell. So this is what we try to identify. So what sort of host proteins are somehow manipulated by the virus protein, and which makes this virus to move, replicate, and etc. And then what is beautiful about this system is if we can identify one or two of these proteins, and we can actually edit using genome editing technologies, those genes, encoding those proteins. And then we will have transgene-free plants.
So the plants presumably need the proteins, but if you can edit them in such a way that it stops the virus, but the plant's still happy.
Yes, exactly. I mean, there are some really good examples as well. So obviously, some of those proteins are essential for plants. There's not much you can do with it. This is why the work get a bit complex in a way. But we can mutate some of those genes, but the protein might still be there, might be doing its function in a way. Or some of them actually just, we don't necessarily see a massive detrimental effect in the plant life, but the plants will be resistant to the virus.
So what technologies are you using? Are you using CRISPR?
CRISPR-Cas9 technology. And what is beautiful about that is you edit maybe a single nucleotide in DNA and remove all the components, and you basically end up with a variety almost identical to the variety you start with a single base pair difference. So in a way, that is really effective, and it will take a long time for the pathogen in general to gain something new to be able to actually infect that particular plant species. Again, evolution in action, really, is the beauty of evolution in that respect.
What's the ultimate goal of your research? What would make you happy if you get to the end of your career and you look back and you go, I did that?
It's not actually an easy question to answer, but for me, if I can see the work that I did with my colleagues in India, if I can see some of those farmers who can't spray their plants with pesticides or fungicides can grow some of those varieties that I'm involved in developing, I think I'll be more than happy, because that means I actually did contribute to global food security. Thinking about future with the climate change, climate change will impact the region where half of the population lives today. Those are the people already actually sort of need support for security. But of course, some of my findings are being used by the next generation. That would make me extremely proud as well.
Volkan, thank you so much for talking with me. This was a podcast by the Milner Center for Evolution at the University of Bath. I'm Turi King, and thank you for listening. If you have any thoughts or comments on this or any other episodes, please contact us through social media. For more information about the Milner Center for Evolution, you can visit our website,